Our first speaker is Arima Genomics' very own Senior VP of Science, Dr. Anthony Schmitt. Without taking too much time here to evangelize and appreciate what Anthony means to Arima, I'll just say a few words. He's the lead developer of the extremely impactful Arima Hi-C kit. He's the lead scientist behind the Arima HiChIP and Capture-Hi-C solutions. He's well-trained, he's well-published in the field, and he's genuinely a good person to work with. Anthony, thank you and welcome.
Thanks, Joe. Can you hear me well? The connectivity is good?
Yep, we got you.
Perfect, thanks. Well, I'm super excited to be here today. I'm even more excited, I think, to hear some of the published and unpublished work that we'll hear about over the course of the next couple hours. I think my function here is really just to introduce the underlying NGS-based approaches for 3D genome mapping and genome structure analysis, and I'll go over a few of those right now. I'm going to first introduce the 3D genome very, very briefly. I know that we'll hear a lot more about that in the talks to follow. I'll talk about our three solutions for genome-wide Hi-C analysis there at the top, as well as a couple targeted versions that we've recently come out with called Capture-Hi-C and HiChIP, and I think each of those are really well suited to address specific targeted hypotheses when it comes to the 3D genome and epigenetics and its relevance in cancer research. So without further ado, I'll get started with the top one there. So first, just a really quick introduction to the 3D genome. This is from a review that my former mentor, Bing Ren, wrote with a couple colleagues in the Ren lab when I was doing my doctoral training there. Here, the idea is just really at the highest sort of level. If you look down at an interphase nucleus, you observe the presence of these chromosome territories, where if you can see my mouse, they're kind of up here depicted as different colors. This is looking down at an interphase nucleus. And this is supported by microscopy data. When you paint different chromosomes, different colors, you see them organizing into their own sort of spatial confines within the nucleus. And then this is also supported by Hi-C data, where the most number of reads or 3D interactions are occurring here across the diagonal in a genome-wide Hi-C matrix. So the majority of the signal is interactions within chromosomes, which supports this idea of chromosome territories. And if you zoom in maybe a little bit further, you see other structural features like parts of the genome or chromosomes tending to associate with the nuclear lamina and other parts of the chromosomes extending more towards the nuclear interior. You see the presence of transcription factories and other puncta that are structurally significant features of genome organization. If you zoom in further on the scale, let's say of one chromosome looking at 1 megabase sort of units, you see these self-interacting chromatin domains here that might be called topological domains, TADs, contact domains. There's a few different terms for them that are somewhat used interchangeably. And these are self-interacting structures that are demarcated by architectural proteins at the boundaries like CTCF. And then when you zoom into kind of on the tens to hundreds of KB scale, you see these chromatin loops depicted here with these frequent point-to-point interactions, really at the anchor of that loop that might be mediated by architectural proteins like cohesin, and they can also bring together cis-regulatory elements like promoters and enhancers and the like. And this concept of topological domains, I guess, is illustrated here in a schematic where you have a linear thread and you have 2 points, A and B, on that linear thread. Now, if the genome was folded randomly and you were to just crumple up this string into a kind of like a ball of yarn, A and B, which are close together in the linear sequence, would also be close together in the 3D space of the genome. But in fact, the folding of the genome is not random. And you'll see cases like this where A and B are actually spatially segregated despite being relatively close along the linear genome. And these self-interacting domains that are depicted here as this model are also often depicted as these triangular structures when you represent Hi-C data. And what does that data look like? When you look at Hi-C data here, this is about a 7 megabase region, this sort of self-interacting domain that I'm highlighting with my mouse would be one contact domain, and then a neighboring contact domain here. And then when you zoom in, like I said, onto sort of a whole nother log order increase in resolution, you see these dots on the Hi-C map, which themselves represent very frequent point-to-point interactions between two regions along the linear genome. So why study 3D genome organization? We're gonna hear some really exciting stories here in the near future, but it's been well, I would say very well studied over the past 10 years or so, just using Hi-C type of techniques alone, but way before that. A couple examples here would be in the context of development. Here's a study by Bing Ren and human ESL differentiation using genome-wide Hi-C. In the context of immunology by one of the speakers here today. In the context of neurobiology from the Akbarian lab at Mount Sinai, and then also in the context of cancer, which we'll hear more about today. And here's a couple studies also from our speakers today, and I think we'll probably hear more about these later on. So getting to the underlying tools, which is what I'm going to introduce. When we talk about the Arima Hi-C platform, let's start with the genome-wide Arima Hi-C Foundation. And it frequently comes up, what are the supported sample types? And there's a lot of supported sample sources shown on the left. I think the most relevant for today is going to be human and mouse. There are another suite of sample sources that are supported. In terms of the sample types, we have user guides for cultured cells, primary cells, whole blood, fresh or frozen tissue. We've worked with OCT embedded tissue. So if you have a specific question about a sample type, please drop that in the Q&A or we can follow up afterwards. And then in terms of the sample amounts, a typical Arima Hi-C reaction might start with about 500,000 to 1 million cells. If you're working with frozen tumor tissue, you might start with maybe 50 to 100 milligrams of that tissue, grind it up, cross-link it, and then probably use a fraction of that for your Hi-C experiment. And then if you're working with, let's say, mouse blood, you might collect 2 to 3 mils of blood, isolate PBMCs or some type of subpopulation from that, and do your Hi-C's experiment on that. And I should also point out that we do have low input protocols available, and I don't have time to go through our low input Hi-C internal data today, but I will point you to one paper that's used our low input Hi-C. Now, really to get into the nuts and bolts here of the workflow, if anyone, if you're familiar with, let's say, ChIP-seq, Hi-C starts in a very similar fashion. You start with your fixed cellular input here, and then while the chromatin is held fixed there within the nucleus, there's really 3 key enzymatic steps that are carried out. First, we digest the chromatin with a cocktail of restriction enzymes. So what is depicted here is essentially 2 pieces of DNA when you're looking down on a cell that are not close along the linear genome, but they are held together in 3-dimensional space by this protein. Once you have your digestion here, those ends get labeled with a biotinylated nucleotide and then ligated back together. So what you started with was 2 pieces of DNA that were just held together in 3-dimensional space, but not actually physically adjoined to each other, now end up cut and religated back to each other. And you have biotin marking the ligation junction point between those 2 pieces of DNA. And this is the most information-rich piece of this molecule where these 2 molecules ligated together. And then you just purify that DNA and you do a quality control checkpoint that we've built into the technology here that is predictive of the quality of the final sequencing data. So it's an experimental predictor of success. Now, I think I've gone through here the sample input requirements. This whole process, which we refer to as Hi-C, is completed in 8 simple steps. The entire process happens in a single tube. You just keep piling on reagents from the minute that you lyse your cells through the final purification of DNA, which is quite nice that there's no spinning or anything that would be susceptible to sample loss. If you're familiar with other Hi-C protocols, we've innovated in using a cocktail of restriction enzymes, which increased the theoretical resolution of the assay and also the coverage uniformity across the genome. This entire process takes about 6 hours, or about one working day to do the Hi-C part. And like I mentioned, this is a predictive QC checkpoint. Now, after this, these molecules here that you purify are a little large. They're about 3 kb. So we shear those molecules, and then we use streptavidin beads to purify the part gray and part blue molecules, which really capture and represent that three-dimensional interaction that was occurring within the nucleus. And then we wash everything else away. We add our sequencing adapters using commercial library prep kits like Kappa, Swift, NEB, Illumina, etc. And then you amplify, and then you sequence these chimeric molecules, and then you map those back to the reference genome. So that will be essentially one data point that, let's say, chromosome 1 and chromosome 4 were interacting within 3D space. I did mention that we have low input protocols. We've gone down to 50,000 cells or less. I would just point you to a recent publication there from the Akbarian lab that I mentioned earlier that had used our kit for Hi-C analysis of 6,000 sorted dopaminergic nuclei that were harvested from postmortem brain samples. And we offer this technology as both a kit and a service format. And I really only have time to show, I think, one piece of data to highlight the benefits of using the kit. And that is really when we first started generating this Arima Hi-C data, we of course set out to benchmark that data against really the gold standard in situ Hi-C data that was available at the time. And that was really an excellent protocol and has continued to be a workhorse in the field. So when we did this comparison, we took some published in situ Hi-C data from 3.6 billion raw Hi-C reads. And all we're looking at here is essentially one point-to-point interaction, which would be characterized as a chromatin loop, about a megabase in size. And when we analyzed the same sample using the Arima Hi-C data, what we observed is that at these looping regions, essentially these structural features that you're often doing Hi-C to detect, that signal enrichment at that looping region was much higher than in the published in situ Hi-C data. And I think the important thing to notice is that signal is more enriched despite there being only a third of the total sequencing depth. So the concept here is just that really in this case in point example, we're highlighting that the signal strength of these structural features is higher despite having less reads than other Hi-C methods. And to take this one step further, what we did is we took the 1.2 billion raw read Arima Hi-C data set and we subsampled that down to 1 billion reads, 800 million reads, 600 million reads, and we called loops using the open source tools called Juicer and HiCups. And I'm just plotting the total number of loops that we find compared to the 3.6 billion read published in situ Hi-C data set. And what we find is just that the number of loops that you can detect is about 50% higher, despite having a sixth of the total sequencing depth. And that's internal data, and our customers have also seen this as well in a different cell line. Again, the concept of being able to detect more loops from less sequencing depth. And there's been several testimonials of those who have used our kit in the field. This is all on our website, and a growing list of publications in the nuclear architecture and epigenetics research fields, which are also on our website. Now, with that introduction really to genome-wide Hi-C, as a company, we've been really, I think, keen to address more targeted tools that are well positioned to address specific targeted hypotheses when it comes to genome organization, gene regulation, and those two tools, like I said, are called HiChIP and Capture Hi-C. And I really only have a time to just barely touch on both of them. And I'll start with Capture Hi-C. I think a good way to introduce Capture Hi-C to those who are not familiar is to position it relative to more popular capture sequencing type of protocols like exome sequencing. So in a typical exome sequencing workflow, you extract your DNA, you shear that DNA, you add your sequencing adapters, and you amplify. And then at that point, before you would sequence, you introduce these biotinylated oligonucleotides that hybridize to the exome, for example, and then you can capture those hybridized molecules using streptavidin beads and enrich your genomic DNA library for the sequences that you want to capture, like the exome. Then you amplify and then you sequence. So that's a typical capture sequencing protocol. Now, the way that Arima Capture Hi-C is different is this process of extracting the DNA is simply replaced with doing Hi-C. So instead of just extracting DNA, you go through the process of digestion, biotin fill-in, proximity ligation, and then DNA purification. Right. So in fact, really, it's the exact same Hi-C protocol that you would do if you were doing genome-wide Hi-C, except right before you would get to the point where you would traditionally sequence, then you add your biotinylated probes and you enrich for any sequence element of interest that you're interested in probing for the 3D landscape. So you can imagine if you want to capture promoters and all of the distal elements that interact with promoters, you would do an experiment where you enrich the Hi-C library for promoter sequences. And we've been partnering with Agilent Technologies over the past year to try to streamline this Hi-C plus capture sequencing protocol, and been using their probes, hybridization reagents, and probe design software suite. So really, I think just to get you thinking about how this might be useful in your research, there's a couple different flavors, I would say, of Capture Hi-C to think about. There's the concept of, let's say you have a gene or several genes of interest, and you're interested in obtaining Hi-C type of data around just those genes and not the whole genome. Then you might do an experiment like region Capture Hi-C, where you design your capture probes to enrich for the contact domain that contains your gene of interest and maybe the flanking domains as well, but then leave everything else behind. There's also been a body of literature that has used Capture Hi-C with the concept being there's lots and lots of GWAS studies that have identified variants that are associated with a disease phenotype, and perhaps those causative variants in the non-coding regions are disrupting, let's say, enhancers, and you want to use the Capture Hi-C data to link those regions to putative target genes that may be associated with the disease phenotype. And lastly, like I said, the concept of promoter Capture Hi-C, where you capture promoters and everything that interacts with those promoters, either at a genome-wide scale or some type of subset of all the promoters in the genome. And the way that we work with our customers for Capture Hi-C is we help really through the entire process of designing the probes. We use the Agilent Sure Design software suite. We have off-the-shelf promoter Capture Hi-C designs for mouse and human. We also do lots of custom designs. We review the design with you on the UCSC genome browser, and we iterate if necessary, and then those probes would be ordered from Agilent. Now, we have a lot of really exciting, I think, internal data. One of those experiments that we've done internally is we've captured the regions around several different select oncogenes or tumor suppressors, like MYC, EGFR, BCR, PTEN, NTRK. And we've done this in a way where, let's say, I'm saying here we capture BCR, 620 kb. What I mean is if the BCR gene is here, we've designed probes to capture the 620 kb around the BCR gene and obtain that high resolution Hi-C data just around BCR. And we've done this in 3 different normal cell lines and seven different cancer cell lines of different tissue origins. All this data that I'm going to talk about here in the future is openly available if you want to have a look at it. So what does this data look like? So this is the K562 Capture Hi-C data. And I want to step through, I think, the real benefit of an experiment like region Capture Hi-C. So let's look at the BCR gene. if you were to take this Capture Hi-C data and analyze it at a large bin size, let's say 25 kb bin size, you would get, you know, something like this, where you have an interaction that you can kind of see that's happening off the diagonal here. And when you trace those back to where those originate along the linear genome, you can see that they're here. It looks like a very pixelated image, and you can't really tell when you look at the chromatin features whether it's CTCF peak here or here or here or here that's associated with this interaction. And you might really want to know whether that interaction is at the promoter of BCR, but you can't resolve that at lower resolution. You can take the same Capture Hi-C data and bin it at 5 kb resolution, but unfortunately, you actually still run into the same issue where if you look at that significant interaction that appears to be happening, and you trace that back to where it belongs along the linear genome, again, you can't really tell if that loop is at the promoter of BCR or just upstream of BCR. You can take the same Capture Hi-C data and cut the bin size by a whole nother log order down to 500 base pair resolution, and now it becomes much more clear that that interaction that you saw when you trace that back to the linear genome is really anchored at the promoter of BCR. So this type of 500 base pair bin size is really unprecedented at the genome-wide scale. It would take on the order of probably, I don't know, 8 or 10 billion reads genome-wide Hi-C to get this resolution. But you can get this type of resolution from targeted Hi-C around your gene of interest for approximately 8 or 10 million reads per locus like this. So for Capture Hi-C, just in summary, we've developed a streamlined Capture Hi-C protocol that combines Arima Hi-C with Agilent SureSelect. We have a full support, completely complementary for probe design and experimental consultation. And here I've showed you how you can get sub-kilobase, ultra-high resolution targeted Hi-C maps around your genes of interest for approximately 10 million total raw reads per megabase of targeted sequence. Now, lastly, we'll go into HiChIP. Similar to how I introduced Capture Hi-C, to introduce HiChIP first in a typical ChIP-seq type of experiment. You have your cross-linked cells, you shear that chromatin, you add your antibody, you do your IP, shown here, and then you do your library prep and sequence. Arima HiChIP is very similar in that it's actually just as it sounds in the name. You start with an upfront Hi-C. So again, digestion, fill-in, ligation. And really at this point, you have this proximally ligated chromatin. Then you shear this chromatin and you add your antibody. So when you structurally look at the molecules that are getting enriched, the difference is that the HiChIP ones, this molecule not only contains the sequence content around the immunoprecipitated protein that you would get from ChIP-seq, but it also contains a 3D interaction information that was associated with this immunoprecipitated protein. So after you do your IP here, then you do your Hi-C style library prep to enrich for the ligation junctions. So in this workflow, there's essentially 2 enrichments, one for the protein and then one for the ligation junctions. And this technology inherently measures the 1D protein localization sites that you would get from ChIP-seq, and then also the 3D interactions that are associated with that immunoprecipitated protein. And lots of the customers so far are using this technology as a tool to characterize chromatin loops that are associated with active promoters or active enhancers. We also have optimized sharing protocols that are compatible with Diagenode as well as Covaris. We've had a really fruitful collaboration with Covaris developing the shearing component of this protocol, and I would just point you to the poster that we presented with Covaris at AGBT this February. And we also offer this as kits or services. Now, why use HiChIP? It's been used in the context of cancer epigenetics a couple times here by the Bernstein lab and Paul Mischel's lab here at UCSD. I would just point you to those papers. when we talk about visualizing the Arima HiChIP data, this is a WashU epigenome browser track around the MIC locus here. So we have our CTCF ChIP-seq data, cohesin ChIP-seq data, et cetera. What I wanted to draw your attention to here when it comes to the performance of Arima HiChIP is the bottom two tracks here are essentially two replicates of ChIP-seq data for H3K27 acetylation downloaded from ENCODE. When you plot the HiChIP data in a 1D signal track, just like ChIP-seq data, the 1D protein localization sort of occupancy data, that 1D signal track in both replicates recapitulates the ChIP-seq data. So you're still capturing those same ChIP-seq peak information around the MIC locus. And it's very similar compared to ChIP-seq. But what you're not getting from the ChIP-seq is that 3D connectivity information that connects these K27 acetylation peaks to other K27 acetylation peaks or promoters or other cis-regulatory elements in the locus that would inform you of a 3D gene regulatory type of interaction. And we've also tried to be thoughtful about making sure that this technology is reproducible in terms of the loops that it calls. So these statistically significant loops that are being identified here using the open source MAPS pipeline appear to be quite reproducible at this locus. And at a genome-wide scale, we find that about 85% of the loops that we find in REP1 are also found in REP2 for the K27 acetylation HiChIP, and a similar degree of reproducibility in our H3K4 trimethylation HiChIP. So the last slide I have here is, I think Arima really prides itself on putting a product out in the market that's been customer validated. And I wanted to go through some of the beta testing data that we've completed before launching the product. So just a couple key performance metrics when it comes to HiChIP. You really want to query the data to make sure that the HiChIP data has a high efficiency of proximity ligation. In other words, it has a strong Hi-C signal in the data, but it also needs to have a high chip specificity signal in the data. So really with Hi-C, you're just looking at what are the fraction of long range interactions that are in the data, and with ChIP, essentially what fraction of the reads overlap known ChIP peaks. So when you plot that data, the percentage of long range interactions in the HiChIP data on the y-axis and the fraction of HiChIP reads that overlap ChIP peaks, you want to be in the upper right quadrant of this plot. And all 24 reactions from the 3 beta testing sites meet that criteria of having a high fraction of long range interactions and a high ChIP enrichment for both antibodies. We then plot essentially what are the total number of reads that are required for at least 70% reproducible loops across the 3 different beta testing sites in many, many different cell lines that were examined across these 3 different sites. And the main takeaway here is that for H3K4 trimethylation, we're needing about 100 million reads to obtain reproducible active promoter-centered loops across the genome. And for K27 acetylation, a bit more of an enhancer-centric point of view, about 300 million reads for those H3K27 acetylation-mediated loops. And if you step back and just sort of think about the sequencing requirements for these types of technologies, for in situ Hi-C, for loop level resolution, I believe ENCODE is recommending about 2 billion reads. For Arima Hi-C loop level resolution, we're talking about 600 million reads. And then for the HiChIP, it's about 300 million reads for K27 acetylation and 100 million reads for K4 trimethylation. I would just lastly direct you to some of the testimonials from these early access partners and beta testers. And in conclusion, for HiChIP, I think we've developed a very nice streamlined in situ HiChIP type of protocol for the efficient capture of both the 3D loops and the 1D chromatin occupancy signal. Those loops are quite reproducible, about 80%. They've been validated across multiple protein factors and cell types. And I didn't have a chance to talk to this, though I'm happy to answer any questions about this, is we have validated multiple open source HiChIP analysis tools, and all this is on GitHub, and we provide extensive technical support, training, and onboarding for the bioinformatics piece. So with that, I'd just like to acknowledge our scientific advisors, Job Decker, Jesse Dixon, and Bing Ren, and the funding support that we've received from the NIH to develop the tools that you heard about today. And if I have any time, I can take a couple questions. Thanks.
Great. Well, thank you so much, Anthony. This is Joe again. Just to keep, in the interest of time, let's keep this moving, but let's take a couple. How about the very first one? Can we use Arima kit on more than 1 million cells? You mentioned the low input capability, but what's the upper level?
Sure. So the upper limit we recommend for the genome-wide Arima Hi-C or Arima Capture Hi-C, it's actually really limited by the amount of chromatin that's in the cells that you put into the assay, because effectively the chromatin that's within the nuclei is the substrate to these enzymatic processes. And we try to work with about 5 micrograms of chromatin within those cells or less. It certainly doesn't mean that using more cells will fail, but the number one cause that we've seen when we try to break the experiment or break the assay is if you put in too many cells, the biochemistry does become inefficient and doesn't run to completion. So I think in short, yeah, we try to aim for, usually it's about no more than 1 to 2 million cells, and then it can definitely go down from there, and sometimes less is more.
Less is more. Great ending. Yeah, that's a helpful hint for those using the kit. Thanks a lot, Anthony. And I'm going to table the rest of the questions. And we have our commercial team on the call, and we will follow up with each of you. We've recorded these questions, but in the interest of time, I'd like to keep things moving. And I'm really excited to introduce our next speaker, Joanna Achinger-Kawecka. Thank you. Okay, there you go. I stumbled a little bit, but Joanna is a postdoc with Sue Clark in the Epigenetics Research Laboratory at the Garvan Institute in Sydney, Australia. Her current focus is on the role of 3D genome organization in cancer and identification of mechanisms by which chromatin architecture arises and is maintained. And I want to say a personal thank you, Joanna, for joining us in the middle of the Australian night. I think it's 2 something in the morning, so we really appreciate that. Thank you for being here. We're all very excited to hear from you, and the floor is yours.
Thanks, Joe. So I'd like to first thank Arima for giving me an opportunity to present my work on the 3D epigenome remodeling in endocrine-resistant breast cancer. So breast cancer is a leading cause of cancer-related deaths worldwide, but around 70% of all breast cancer patients are estrogen receptor positive. And these patients will receive some kind of endocrine treatment. And as you can see on the Kaplan-Meier plots, this treatment significantly reduces the risk of relapse. However, there's still around 30% of patients, I'm sorry, close the chat. Yeah. Around 30% of these patients treated with endocrine resistance will relapse within the first 15 years of receiving treatment. And that is due to development of endocrine resistance. And our group has a long-standing interest in understanding the mechanism behind endocrine resistance, with a special focus on the epigenome. So in order to look at the epigenetic changes associated with endocrine resistance, we use a cell line model, which consists of ER-positive breast cancer cells, so MCF7s, which are then treated for long-term with either tamoxifen until the development of a tamoxifen-resistant cell population, which we call TAM-R, or with another endocrine treatment called fulvestrant until development of fulvestrant-resistant cell populations, which we call FAS-R. And using this experimental model, our group has previously shown that when we look at the DNA methylation patterns in endocrine resistant, shown here in red, and endocrine sensitive breast cancer cells, we can see that there's huge hypermethylation in the resistant, in the two resistant cell lines, and that this hypermethylation is associated with loss of ER binding, and that frequently occurs at enhancer regions. So considering this hypermethylation happens at enhancers, my hypothesis when I joined the lab was that loss of ER binding at these enhancer regions, as shown here, which is due to hypermethylation, results in a disruption of local enhancer promoter interactions and then aberrant gene expression in the resistant cell populations. So in order to study this, in order to validate this hypothesis, we'll use in situ Hi-C in the endocrine sensitive MCF7 cells and two endocrine resistant cell lines, TAMAR and FASAR. And we looked at changes in 3D chromatin organization and then compared that to different histone modifications and transcription factor binding sites using ChIP-seq. We also looked at DNA methylation using whole genome bisulfite sequencing, and we looked at changes in gene regulation using RNA-seq. So as Anthony already mentioned, the 3D genome is organized inside the cell nuclei in a very tightly organized manner. And the first level of organization that we looked at were the chromatin interactions. So these are interactions frequently between enhancers and promoters. They are maintained by CTCF and cohesin, and we know that chromatin interactions connect regulatory elements like enhancers to the target gene promoters, and that facilitates correct gene expression patterns. So in order to identify differential enhancer promoter interactions between the sensitive and resistant cell lines, we used a diff Hi-C package. And using this, we identified around 2,000 differential interactions between the MCF7 and TAMAR cells, and around 5,000 differential interactions between MCF7 and FASAR cells. Interestingly, we saw not only big differences between the parental MCF7 cells and the resistant cell lines, but also there was high dissimilarity between the TAMAR and FASAR differential interactions, with actually just a few common differential interactions between them. We observed both loss of existing interactions, shown in green, as well as gain of novel interactions. Yeah, and as I mentioned, there's only a few common differential interactions between the TAMAR and FASAR. So we obviously next wanted to see where these interactions are happening in relation to the epigenome. And we saw that gained interactions were highly enriched for active enhancers and promoters. Shown here, when you can see that lost interactions have lower enrichments compared to the gained interactions in TAMAR and FASAR. Considering these are active enhancers and promoters, we looked at the expression of genes located at anchors of these interactions, and we observed that at anchors of gained interactions, majority of the genes were upregulated, while at the anchors of interactions that were lost in resistant cells, we saw downregulation of gene expression. And there's still a lot of genes that go in opposite direction, which we haven't really looked at. So here is an example of a GREP1 gene. And you can see in that gene that all those long-range interactions that are present in the endocrine sensitive cells here are lost in both of the resistant cell lines. And this loss of interactions is associated with complete silencing of gene expression, as shown here on the RNA-seq track. And importantly, GREP1 gene is a very well-known predictor of endocrine resistance, and its loss is associated with worst patient survival in endocrine treated breast cancer patients. So a key gene that we saw is being downregulated in endocrine resistant cells is ESR1 gene, which encodes the estrogen receptor itself. So here you can see that its expression is significantly downregulated in TAMAR cells, and it's completely lost in FASR cells. And we know that estrogen receptor binding is significantly remodeled in resistant cells, as well as in clinical samples. When we compared the actual HiChIP-seq data between the MCF7 and TAMAR, we saw that there's a huge loss of ER binding and some, as well as some gain of ER binding. So next we looked at the association between the differential interactions we identified and altered ER binding sites. And we saw that loss of ER binding was associated with loss of interactions in tamoxifen resistant cells, while gain of ER binding was associated with new gained interactions in tamoxifen resistant cells. Actually, when we looked at the total overlap, we saw that around 30% of both lost and gained interactions were actually ER-bound differential interactions. And when we looked at the gene ontology terms enriched at this key ER-bound differential interactions, we saw lots of breast cancer related pathways that have been previously associated with endocrine resistance. So our previous work has shown that DNA methylation at enhancers that are bound by ER is associated with endocrine resistance. And so to confirm this result, we now looked at the genome-wide DNA methylation patterns, and we again observed that hypermethylated, differentially methylated regions are associated with loss of ER binding in tamoxifen resistance, while hypomethylation was associated with new ectopic ER binding. When we plotted the DNA methylation data genome-wide at the ER binding sites, we saw a big shift in methylation at these sites. So less methylation at new gained ER binding sites in tamoxifen resistant cells. And importantly, when we looked at ER binding sites that were gained in endocrine resistant tumors from the public data, we saw less methylation in those metastatic tumors in whole genome bisulfite sequencing from metastatic tumors as compared to primary tumors. However, we only have a couple of tumor samples to look at. So this association is not significant, but we're looking at getting more samples soon. So this is an example of a region where you can see loss of ER-bound interactions in tamoxifen resistant cells, which is associated with DNA hypermethylation. So again, in MCF7 cells, you can see those big long loops at the NCOR2 gene locus. And these loops are completely lost in the tamoxifen resistance. And if you look at the location of anchors of this long lost loops, you can see that this is associated with loss of ER binding in tamoxifen resistant cells at all of these loops. And if you zoom in into those methylation at these locuses, you can see that in each of those differentially methylated, you can see differentially methylated regions at these anchors. So here on those zoom in pictures, you can see DNA methylation track for the MCF7 and TAMAR cells, as well as the whole genome bisulfite sequencing from primary and metastatic tumors, when you can also see this hypermethylation occurring, at least in some of the samples. We also looked at another layer of 3D genome organization, which is the A and B compartments. So these are chromosomal compartments that are either active or which we call compartments A, or silenced. And this, yeah, so compartments A are associated with high levels of transcription and open chromatin, while compartments B are usually located on a periphery and are associated with gene silencing and closed chromatin. So we looked at the association between compartment changes and differentially expressed genes. So we found that around 2% of compartments switch between either from A to B or B to A between the sensitive and resistant TAMAR and FASR cells. And we actually saw similar number of A to B and B to A changes in the resistant genomes. We observed that these changes in compartments was significantly associated with a change in gene expression, where we saw that if a compartment changed from active A to inactive B, we saw a general downregulation of gene expression. And if a compartment changed from B to A, we saw upregulation of gene expression. So here is a visual example of such a region that changes from active in MCF7 cells, shown here, to inactive B compartment in TAMAR and FASR cells. And this change is associated with loss of expression of multiple genes within that region, as shown on the RNA-seq track. So we also wanted to see what's the association between the compartment structure and the ER binding profiles. And we saw that if a compartment changed from A type to B type, we saw general enrichment for lost ER binding sites in resistant cells. And when the compartment changed from B type to active A type, we saw enrichment for gain of ER binding sites. And again, just to show you a representative example of that, here is a region in MCF7 cells that's inactive and compartment B. That region becomes active A compartment in TAMAR cells. And on the ER-ChIP track, you can see a huge increase in ER binding at that gene locus. So in conclusion, we have shown that 3D epigenome remodeling is a key mechanism underlying endocrine resistance in ER-positive breast cancer. And that includes differential ER-bound enhancer promoter interactions, which are associated with aberrant expression of key breast cancer genes. They're located at altered ER binding sites in resistant cells, and they are associated with DNA hypermethylation at these sites. Additionally, we saw that a change in A and B compartment structure, which occurs at differentially expressed genes and at altered ER binding sites. And with this, I'd like to thank my lab with PI Susan Clark at the Garvan Institute and all the people who contributed to this work. Thank you.
Thanks, Joe. We have several questions here. Let me start with Shara Lampos asks, what causes the changes in DNA methylation at the sites that you presented?
So I would think that these sites are not being, so we know that ER binding changes in that the ER can be kind of shuffled or pushed around on the genome to different sites to activate different genes. And we think that DNA methylation is kind of the final mark that closes the chromatin at these sites and makes it inaccessible, and that's why we lose the looping. So it's kind of a closing mechanism for the sites.
Okay, yeah, I think that there was a related question to that that maybe you touched on a little bit is, Papudu asks, can you say whether these changes and interactions that you observe are causal, or are they results of other dynamics like DNA methylation or histone modifications?
Yes, I think that's a very good question. So we would like to think that it's possibly that the epigenome has to change first. So either the DNA methylation has to be gained or lost, or the chromatin, like the accessibility, has to be changed for the loops. And we do actually have a temporal model of endocrine resistance in which we plan to check what actually happens first. But I think everything happens very, very fast, which means that we would need very fine temporal data to be able to tell whether it's gene expression that happens first, accessibility, DNA methylation, or looping. But yeah, that's definitely something I'd like to check.
Okay, thank you. Sabria asks, do you have evidence of estrogen receptor associating with mediators of promoter enhancer looping, like mediator, complex, cohesin, or at least sharing common binding sites?
So I haven't looked at it myself because I don't have the cohesin. I have cohesin data for the cell lines, but it's not as, so we don't get many peaks, and it's not very good quality, and cohesin is quite difficult to chip. I know that ER and promoter enhancer looping is associated with different chromatin remodelers, which has been shown previously by Jason Carroll's lab. But I haven't looked exactly at cohesin and the association. I think that's mostly because it's not such a good chip as CTCF, for example.
Okay, there's another question here that I guess I was thinking of as well. Jihad asks, have you validated these types of findings in primary tumor tissues?
Yes, so this is where our collaboration with Arima comes in. So we do have endocrine resistant PDX tumors, which were implanted in mice. We have a couple of different PDXs, and we are currently working with Arima to use their low input method on fresh frozen tumor tissues to see if we can see the same kind of changes, at least in PDXs for now, because that's where we have most of the tumor tissue available. But if we get the method working, we would like to move to actual patient tumors, which we do have for our collaborators at the King Kong Cancer Center in Sydney.
All right, maybe just one or two more technical questions about the analysis itself. Subhashri asks, how do you identify different compartments A and B from the Hi-C data?
So I know there's a couple of different methods to identify compartments. For this work, we used Homer, which was the easiest one because it just works on mapped reads files, or BAM files. So these are just PCA kind of analysis, but I know that Juicer as well has now eigenvalue compartment calling. So there's a few different methods. Yes, but in this work, we used a software called Homer.
Okay. I think we'll wrap it up there. And I guess, Joe, would you like to introduce the next speaker?
Yes, sir. First, I'd like to say thank you so much, Joe. And please do not allow your little ones to wake you up as you sleep in. We appreciate it, and get some rest. We'd like to welcome our next speaker, Iannis Aifantis. Iannis is a professor and chair of the Department of Pathology at NYU Langone. Among other great things, Iannis is a cancer researcher focusing on acute leukemia. He's got a fantastic lab who are a pleasure to work with and are focused on the molecular mechanisms driving normal stem cell differentiation and malignant transformation. We're extremely pleased to have him here today. Welcome.
Hey,
Yeah.
Joe, I hope that you hear me. Thank you for putting this thing together, and it's really exciting to see so many people popping up here. I guess that's probably the only good thing that happened from the COVID epidemic or pandemic, to see all these people basically participating in science events like this one. So I will tell you about a part of the work that we're doing in the lab on 3D organization and leukemia. I'm gonna skip the first slides because you have introduced them as a first speaker so well, so I don't have to do it. We are going to talk about TADS and looping organization in leukemia. And as I'm going to show you later, we have done a lot of Hi-C and HiChIP analysis and also 4C analysis that we're going to mention to you later. The way that we got into this particular question is through work that we have done in non-coding genome and non-coding RNAs. And one example is that work we have done probably more than 5 years ago now, identifying link RNAs that could actually hold together enhancer and promoter areas. And we characterized one of them that we call LUNAR1 in T-cell leukemia. Subsequently, we were very interested in basically boundaries of TADs and structural proteins for DNA looping. And we have generated mice that lacked several members of the cohesin family, specifically in hematopoietic cells. And we have shown that these mice basically develop myeloid neoplasms and acute myeloid leukemia. And basically one thing led to another. The question that I would like to address today is a very simple one. The previous speaker somehow touched into it, is basically, are cellular transformation events and cellular transformation itself accompanied by changes in 3D chromosomal organizations? We are using T-cell leukemia as a model. A few words, T-cell leukemia is a pediatric disease mainly. It can be also found in adults. And as you will see there later, T-cell leukemia is driven mainly by transcription factors like Notch and MYC, and can be broken down to different subclasses. One of them is, the larger one is this classical Notch-driven T-cell leukemia, and the smaller one is something that is called early thymocyte progenitor, or ETP ALL, and I'll show you later why that's important. This work has been done by Raja and Andreas. Raja has done all the biology, Andreas has done all the computation, and actually Andreas is with us today, so he can help me with addressing questions, because fortunately or unfortunately, I'm not doing experiments by myself. And all of this, of course, could not be done without Aris Tsirigos, who is one of the pioneers in the computational analysis in this particular field. So what we've done, very simple experiment, we got either peripheral T-cell leukemia cells from patients that have TLL, or normal T cells or progenitors. And we have done in situ Hi-C, HiChIP, 4C, RNA-seq, and a lot of ChIP sequencing for things like CDCF, H3K27 acetylation, and a couple of other things that I'm going to show you later. And the analysis was done using the Hi-C bench that Haris Lazarus, who is also on the line, and Aris Tsirigos have pioneered in our own institute. So basically, the first thing that we have seen is that if we take unsupervised and we just do an PCR clustering unsupervised, basically measuring Hi-C boundary scores, we can see that we have 3 different categories of cells here. And I'll call them the blue ones, the green ones, and the red ones. And if we're breaking them down based on the identity of the cells, hematopathology data, and also FACS data, they break exactly like that. So up there we can see what I called a conventional standard T-cell leukemia. On the right here of the PC1 is normal T cells. And down there we can actually see what we call ETP-ALL, which is basically, as I've told you before, an an earlier in differentiation subclass of ALL. That now, and this is unpublished, basically poses the question, what is the origin of all of these subtypes of ALL? So now what we're doing in the lab is that we are basically combining a lot of sequencing of primary and normal stem cell and thymocyte and T cell subsets and compare them with different types of ALL. So we have done a lot of analysis, and you can take a look at the paper. Today I'm just going to tell you two stories. One is basically on this idea of intra-TAD activity, and as you can see here, we can have these changes between a TAD that is active, that TAD becomes more inactive during transformation, and of course the opposite. And if you look at that and compare T cells and TLL cells, you can see a lot of these genes that appear to be inactive TADs, and some of them are kind of like classical oncogenes in T-cell leukemia, and also are genes that are driven by Notch and MYC as we actually expected. And you can see comparison down here of two normal T-cell samples, and you can basically don't see these significant changes that we see above when you compare normal T cells with T-cell leukemic cells. Very quickly, we have seen that this intra-TAD activity changes correlates very nicely with differential expression of RNA, and as you would have expected, correlates with CTCF insulation and strength of CTCF binding. The other thing that we have seen is that we do see, as you can guess based on the PC data that I've shown you before, differences between canonical or classical TLL with ETPLL. So we can see, for example, in ETPLL that is more early disease and depends on things like expresses more myeloid markers and expresses and depends on FLT3 activity. We can see higher activity of a TAD that includes FLT3, and this is the example here. You can see the higher activity in ETPLL, and this is the sum of TAD activity that correlates very nicely with high levels for FLT3 in RNA sequencing. On the flip side, we can see that Notch targets, like this gene that is called ABCDD1, is a Wnt inhibitor, and a classical Notch target, we can see higher TAD activity and higher T-cell leukemia, RNA sequencing expression in conventional T-cell leukemia. What I kind of wanted to focus today is these differences on TAD insulation and the changes that we've actually seen in T-cell leukemia, and start with this idea that we had some of these examples. We could have, for example, what we call a TAD boundary loss and a fused TAD, or exactly the opposite, basically a TAD boundary gain and a gain of a TAD boundary with a CTCF site, and the schism that happens leading to basically two smaller TADs. The first thing that we've actually seen is that we have seen quite a few genes that appear to be tumor suppressors and oncogenes that actually follow these patterns. And one of them, and I'll focus on that, is actually MYC. And I've told you before that MYC is one of the drivers in oncogenic drivers in T-cell leukemia. So what we've seen before, and this is data that came from Francois Spitz and Adolfo Ferrando, is that there's a MYC enhancer, which is in the whatever people used to call gene desert, basically downstream of MYC, that controls expression of hematopoietic MYC gene. And also in T-cell leukemia, this enhancer is basically driven by Notch binding, and if you abolish Notch binding, you basically lose the activity of this particular enhancer. So let's take a look at this locus, and sorry that is a busy slide, but we can go down there and we can basically see that in normal T cells, we have two different TADs that fuses in T cell leukemia. And you see this very strong interaction that we can see in primary T cell leukemias and also in cell lines that come from T cell leukemia patients. And that happens because this strong CTCF binding site that you see in T cell basically disappears, and you get this like fusion event between the two smaller TADs. You can see the enhancer down here, what we, Adolfo Ferranto called NME, and a downstream enhancer characterized by the Spitz group. So this, let's call it super enhancer elements here. They don't exist in normal T cells, but they do exist in T cell leukemia. And you can see them here with K27 acetylation, attack sequencing, and you can appreciate notch binding in these particular enhancers, pretty strong notch binding in all of these elements. We can see this interaction basically using FISH, and you can see normal T cells, look at the cell separation between MYC and the enhancer. And in T cell leukemia, cell lines and cells, you can basically see this fusion event and the getting together of enhancer and promoter elements. And of course, that leads to a much higher expression, which is transforming for these cells. So to further test this interaction, we've also done 4C. And you can see here, let's compare, CATL1 is a TLL line in blue, and you can see this significant interaction on the super enhancer with the promoter of MYC. And in T cells, you see basically some interaction with this PVT1 non-coding RNA that has been characterized before. The interaction kind of stops abruptly where the CTCF site is. And then after that, we don't see any more interaction with the downstream enhancer element. We can do the same thing with actual TLL patient samples and not cell lines. And although, of course, it's more noisy because each person is different and each patient is different, you can obviously see these huge interactions with the super enhancer elements. And again, the T cells stop basically at the CTCF site where the first TAD basically ends. The next question was, that's fine. We have interactions between MYC enhancer and MYC promoter, and we have interactions that differ between TLL, that is notch driven, and normal T cells. Can we quote unquote correct aberrant 3D landscapes by drugs? And we have used initially a drug that basically targets a signaling pathway, and this signaling pathway is of course notch. And here we're using a drug that is called gamma secretase that blocks basically cleavage of notch from the surface. So if you cleave notch from the surface, it stays there, it's not active anymore. This drug is actually very efficiently at switching off notch signaling, but unfortunately years ago it failed in TLL clinical trials because of effects of the drug and of course notch signaling in other tissues and specifically the GI system. So what happens when you add gamma secretase inhibitor? You get pretty efficient loss of K27 acetylation and enhancer activity. And this is very specific, the loss of H3 K27 acetylation for genes that are basically bound by notch and dynamically controlled by notch signaling. And there's a pretty nice correlation between loss of these interactions with loss of looping events in genes that are classical notch targets like APCCDD1, as I've shown you, or the IGF-1, LUNAR1 locus that I've described in one of my first slides. So let's take a closer look to some of these examples. So the first one is IGF-1R. You see it here. And this is the long non-coding RNA LUNAR1. And there's an interaction between the two of them. So basically, LUNAR-1 interacts with an enhancer or intronic enhancer on IGF-1 R, and you can basically see the interaction here. If you add gamma secretase inhibitor, you go from black to blue to green, and you basically lose this particular interaction. If you look at a different gene, which is this Wnt inhibitor APCCD1, also not controlled, you can see a very nice also loss of interaction on the enhancer that controls expression of this gene. However, not all these genes are the same. Some genes and some loci are basically immune to treatment with this particular drug, and MYC, it's one of these examples. So you see here, this is 4C data. You basically see interactions between the MYC enhancer and MYC promoter, and you can basically see that you can add gamma secretase inhibitors. You don't touch really the interactions. You decrease significantly gene expression for MYC. You somehow decrease K27 acetylation, but you don't decrease looping. And of course, one of the first questions that we've asked is there a correlation between loss of K27 and loss of interactions? And I would say, although we don't come out and boldly say that in the paper, we usually do see this kind of correlations. So we see that the enhancers that are particularly targeted by this kind of drugs and affected by these drugs, looping is also affected. So of course, the big question I'm gonna stop with this set of experiments is what protects some enhancers from notch inhibition. So I've shown you that in the case that you have notch sensitive enhancers, you have a notch binding. You add GSI, gamma secretase inhibitors, you lose this binding. And then you have K27 acetylation on, let's say, on an enhancer that you lose the enhancer, and that of course impacts looping. And on the flip side, you have notch insensitive enhancers in which you lose notch binding, but you don't lose K27 and you don't affect basically looping in this particular area. So to address that, we've actually used publicly available transcription factor binding data in T-cell leukemia, and we've asked the question, what basically differentiates sensitive and insensitive enhancers? And what we've actually seen is that we don't see differences for notch itself or its cofactors like RBBJ. We looked at BRD4, we didn't see any differences there. We looked at MED1, we looked at P300, but what makes a difference apparently is the binding of CDK7. So what is CDK7? And this is something that we've seen in multiple loci, obviously, and I'm just going to show you a couple of examples. I think I'm going to show you today APCCDD1, MYC, Icarus, and Deltex. So what is CDK7? CDK7 is a CDK, is a cell cycle dependent kinase, but it's a transcriptional one. So it's very important for the initiation and start of the elongation of RNA pol 2, and of course transcription itself. So CDK7 is kind of one of the first post-translational events that happen on the tail of RNA polymerase 2, and that basically pushes to the next step, which is RNA pol 2 elongation. Several groups, including Nathaniel Gray's group at Harvard, have actually developed CDK7 inhibitors, and they have actually tested them in T-cell leukemia in the same model that we are using, and they see a pretty nice in vivo and in vitro response. So when we've seen CDK7 on these enhancers and promoters, we got particularly interested because we knew the data on the effect of CDK7 inhibitors on this particular disease. So what we've done is that we've used the Nathaniel Gray inhibitor, THZ1, And basically what we've seen is that, of course, CDK7 inhibition affects globally HCK27 acetylation. In most cases, we do see loss of K27 acetylation. We do have gain in some areas. We see that basically enhancers that are sensitive to this inhibition are now basically rewired the binding and looping with nearby promoters. And we do see that enhancers that are affected and they're losing K27 acetylation and their activity in response to CDK7, they basically lose looping strength in these particular areas. And I'm just going to show you a couple of examples. Actually, one example. It is, again, MYC. Remember what I've told you before. Inhibition of Notch itself is not enough to change looping. However, CDK7 inhibition really disrupts MYC superenhancer interaction, and you can actually see it here. This is on Jurkats, but we've done it in multiple cell types right now. You see this very significant loss of K27 that is accompanied by loss of interaction between enhancer and promoter, or enhancer elements and promoter. And this is something that we've seen on Hi-C, HiChIP, and 4C. So basically what I've told you today, and that is published, and you can go and get this week more information on the actual paper, that we believe that cellular transformation is accompanied by changes in 3D chromosomal landscapes. And this is what the speaker before me also told you in the breast cancer system. I've told you that 3D organization changes is a novel mode of altering activity of oncogenes and tumor suppressors. And this is something that particularly interests us because, you know, it might be a mutation, it might be a changing in methylation, it might be a deletion or a loss, but it could actually be just a change of 3D organization that makes a tumor suppressor a tumor suppressor. And the last thing that I wanted to mention is that I've shown you some first experiments that show that drug treatment can correct aberrant 3D organization. But we believe that drug combinations are more efficient in achieving that. So, for example, combining a drug that targets a specific pathway and maybe a more transcriptional or epigenetic regulator like CDK7 could be an interesting one in this particular disease that I've talked to you about. So I'm going to stop here. The work was done by Andreas and Raja, as I've told you before, and was supervised by myself and Aris Tyrigos, and we could not do it with several people in the lab, without several people in the lab, Sophia, Harris, Joanna, and Panos, who is now at Northwestern, and a lot of collaborators around the country and the world, and a lot of thanks to Arima, because we've used their kits for, I would say, the majority of these experiments. And I'm gonna stop here.
Excellent, thank you, Iannis. We have a few questions here. The first one is from Xiao Ten. Why do you think you observed the CTCF loss? I think probably referring to the earlier data at the MYC locus, maybe DNA methylation. Is there a mutation in the binding site or something else?
Yeah, so this is a great question, and I think we spent months, not even a year, maybe even a year on that. So we tried methylation. We don't see any changes in the methylation in that particular area between normal and tumor. We have tried sequencing. So we've done sequencing of the area. We don't see any mutations. We have played with a lot of different things. The short answer was we don't know. The longer answer is that one thing that is different is that if you look at attack sequencing tracks in this particular area, you do see open chromatin basically in T cells and closed chromatin in TLL. So we believe that there is a change on basically chromatin accessibility that might define the binding of CTCF in that case, but which is the factor that actually does that, we don't actually know at this point.
I think a related question I had, just to maybe follow up on that, is when you treat with the CDK7 inhibitor, do you see any evidence of the CTCF binding coming back at the MYC locus?
We have tested that. CTCF binding is not coming back. So you do see a loss of interactions between the promoter and all this compendium of enhancers downstream, but we do not see CTCF binding coming back.
Okay. A question from Jihad. When you use the CDK7 inhibitor and you're impacting lots of interactions, what's the impact on cell identity? Are the cells differentiating at all?
That is a great question, and this is work that we are doing currently in diseases like myeloid leukemia that depends more on differentiation. We did not see change in cell identities. The problem is that TLL and T cells at this point is kind of terminally differentiated, so it's very difficult to see changes in cell identity. We should have maybe cytospin the cells and try to see if we have these kind of changes, but morphologically and by fax, we didn't see any changes.
Okay, thank you. A question here from Bogdan. Is the TAD calling or the TAD differential analysis impacted at all by underlying copy number variants or structural variants in the ALL cell?
That is a great question. That's another month-long work that Andreas and Aris have done in the lab. So we have been through that. We have filtered out any of this kind of translocations. We were lucky that TLL, pediatric TLL, is not particularly unstable as a disease. So it was relatively easy for us to basically filter out these few translocations that existed in each sample and make sure that whatever we define as interactions are not affected by this kind of chromosomal events. But I absolutely agree. There are tumors, both leukemias and specifically solid tumors, that people have to be very careful with their analysis.
All right, one more question, I guess, about the analysis methods. Can you say more about the TAD activity metric and just how it's defined?
So, I mean, Andreas, which is on the line, could actually write an answer if you want, which is more sophisticated than mine. I mean, the way that we do it is that we calculate interactions within the TAD and in the boundary area and in the TAD that is adjacent. So basically, it is a computational way to quantify interactions within the TAD and also quantify and define the insulation and the loss of interactions that happen on boundary areas. Take a look at our methods and maybe even Andreas can help with that.
Okay. All right. I think, let me just do one last scan here. I think that's actually Andreas wrote in. Let me just read it out loud. So the TAD activity metric is the sum of the interactions within the TAD area. It was initially a mere technical reason when we had sequencing depth as low as 150 million total reads. To get an answer to regulatory loops, we use the sum of the interactions as an approximation of such interactions, which is associated with RNA-seq and notch binding. So that is a very quick description, and like you said, to look into the nature of genetics methods